All right, everyone, thank you for joining us today. Thank you for spending your Friday afternoon with us or morning or evening, wherever you're at. My name is Karen Marisi. I wear many hats in this community. The most important hat, though, and the most stressful is being a mom to a little boy, Dominic, who has PMM2 CDG. I also am the patient and community outreach coordinator for CDG Care, and I've had the privilege of working closely with the Glycomine team over the past few years, really helping to make sure that their approach is always focused on the needs of patients and families. The real challenges that families like us face every single day. I first learned about glycomine and this trial back in 2018. So that kind of gives you an idea of just how long this journey has been and how many moving parts have had to come together to get here today. The enormous amount of effort it took from the glycomine team to be here, and the amount of dedication. So thank you. I know that there's been a lot of conversations and chatter within our community about some of the details surrounding GLM 101. So that's why we're really excited to have this opportunity for you, for everyone that is interested, so you could hear directly from the experts. So we ask that you please hold your questions till the end. We will have more than enough time to get them answered. And without further ado, I will introduce our two presenters for today. Joining us, we have Dr. Rose Marino, who began her career as a pediatric endocrinologist and later moved into drug development in the rare disease space. She now serves as Chief Medical Officer at Glycomine, where she oversees the clinical program and leads efforts to bring this potential new treatment forward. And we're also joined by Dr. Andrew Edmondson, who serves as a co-principal investigator in the National Network of CDG Specialists and Researchers. Dr. Edmondson brings a deep understanding of many CDG types and is actively leading research to advance new treatments and therapies. In addition, he is the founder and director of the Edmondson Lab at the Children's Hospital of Philadelphia, where his team focuses on improving care and outcomes for individuals living with CDG. Thank you both for taking time out of your busy schedules to be with us today.
Great.
Thank you.
Thanks, Karen. And thanks, everyone. I echo Karen's sentiment. Joining us today, taking time out of your busy schedule to hear about our trials that are both ongoing, which we'll share some data today, as well as what's coming now in the future. So we will discuss three major topics today. So Dr. Edmondson will go into a bit of PMM2-CDG, which I know everyone is probably well versed in, but just to sort of set the stage. I'll take it over and share a bit about Glycomine's journey, bringing GLM101 into the clinic, as well as discussing some of our interim preliminary data from our ongoing phase 2A trial. And then Andy will discuss then where we are now in terms of the new trial, the GLM101-003, or the POLAR trial, and the status of that, and discuss a little bit more of the details. And after that, we'll open it up to questions that we're both happy to answer in regards to the data, the trials, or even just PMM2-CDG. So without further ado, Andy, I'll drive the slide, so I'll hand it over. Yep.
Sounds good. So yeah, just to set the stage, to review what is PMM2-CDG and why is GLM 101 an exciting sort of therapeutic approach to treating this disorder. So, glycosylation is this very basic process where we add sugar groups, so these sugar trees, glycans, to proteins. Those proteins require those to properly function. And in CDG disorders like PMM2-CDG, that process is disrupted, and so the proteins are missing their glycans or may have abnormal glycans attached to them, and those proteins are no longer able to properly function. So this is the process that the body uses to take mannose, one of these sugars, and turn it into a building block so that it can be used to build those glycans and glycosylate the proteins. And you can see that PMM2 in that sort of orange oval structure is really important in taking this mannose 6-phosphate form, turning it into mannose 1-phosphate. And so it really plays a very central role in being able to generate the mannose for the glycosylations. And if you don't have PMM2, because you have a condition like PMM2-CDG, then you can't do that. And so you will not be able to take these precursors for the glycan and turn them into the mannose that can be used to build the structure. And so you get a deficiency or lack of the mannose 1 phosphate in the body. And that's where GLM101 comes in. So GLM101 is designed to restore that mannose 1 phosphate that PMM2 isn't generating in patients with PMM2-CDG. So it will restore that mannose 1 phosphate. So GLM101 is mannose 1 phosphate, but you can't just get it straight into cells. And so in order to get it delivered to the cells in the body, and to protect it from being broken down before it gets delivered to the cells where it's needed, it's encased in a liposome. And that liposome then surrounds it, protects it, and helps with its appropriate delivery. And that's what GLM101 is a liposome encased mannose 1 phosphate that's delivered by an IV infusion once a week.
Great, thanks. Thanks, Andy. So I'll take it over from here, and we'll go a little bit into what Glycomine has been up to, as well as the results before we talk about our ongoing now phase 2b trial. But even before we get there, just to set the stage on what it takes to bring a potential therapy into the clinic so that Dr. Edmondson and all your physicians can write a prescription for a treatment for a disease. It takes, as Karen said, it takes many years and a lot of time and effort in multiple different steps that lead up to that final approval. So initially, and it's not even on this slide, it starts even before you get into the clinic, where you have a drug candidate, you would think about it and test it in cells. You would bring it into animal models, both small animals like mice, then bigger animals to think about how it reacts in the body of an animal before, and safety before you would even think about bringing it into a first-in-human study, which is that phase 1 study. A lot of times for diseases such as PMM2-CDG that are rare, natural history studies are very important to help understand how the disease progresses over time and what sort of markers or clinical endpoints could be studied with a potential therapy that could positively impact patients' lives. So a lot of times you'll have a natural history study happening really in parallel to some of these early phase development studies, both preclinical and then earlier in the clinic. So a phase 1 study happens, that's that first in human, after you've done all of your work in the preclinical stages. You've seen where you see good safety in the animal models. You bring it into usually typically healthy volunteers. So sometimes you can bring it into to patients with the disease before going into healthy volunteers, but the typical way it's done is you've got a small study in healthy volunteers. You learn how the drug reacts in the body and how it metabolizes, how the safety looks in those that are not having the disease of interest. Once you've done that, you then bring it into what we call phase 2, where you treat a small group of patients with the disease. You're still looking at what the best dose you would want to bring forward, as well as endpoints, and of course, always safety. And then from there, once you see that your potential therapy is safe and you've got some good evidence to show that there's some efficacy that's helping patients with the disease, then you bring it forward into a bigger phase 3, or what they call a pivotal study, which confirms the efficacy. And usually this is done by comparing it to a placebo, but phase 2 studies also can typically have a placebo arm to compare as well. And then from there, you bring all of this information together from your preclinical, through your natural history, through all of your phases of development. You put it all together, you present it to regulators such as the FDA or EMA, and you see how the data comes together for a final approval, looking at the risk benefit of the products that you're looking at for a particular disease. So in terms of Glycomine and GLM101, what have we done? Thinking about this roadmap, we have conducted a natural history study. Greater than 3 years, we have some patients with about 5 years of data and about 140 patients with PMM2 CDG. This study is ongoing now. It's finishing up. We're sort of at the tail end of this natural history study. It will be finishing up in about January, February of 2026. We've had a completed phase 1 study, as I discussed, that was in healthy volunteers, which allowed us to determine the dose to bring into the phase 2 study, the 2A study, where we are treating patients with GLM101 with PMM2 CDG. This study's been ongoing for a couple of years now. It is actually finishing up in about a month. So this study has been completely enrolled and has been for about 6 months, and now it's going to be completely finished in a month or so. And this is the data that we'll discuss over the next few slides that got us very excited to continue to look at GLM101 as a potential therapy for PMM2 CDG. And that led us to the phase 2B study, the polar study that Dr. Edmondson will talk about after I go through some of the data from the 2A. So the GLM101 was used in an open label study called 002. So what that means is that everybody knew they were getting GLM101, both the clinicians, and the patients, as well as us as the sponsor. The design was a 6-month IV weekly infusion of GLM101. We treated 27 PMM2 patients from as young as age 3. The entry criteria, you had to be older than age 2 or up. Our youngest patient is down to age 3. And today we'll share some of the interim results that we have from those patients from the teenage years as well as the adults, as those are completed patients at this point. And we're still looking at finishing up the study, as I mentioned, and gathering all of that information. And we'll be able to report on the whole cohort in the next couple of months. The primary endpoint for this study, as well as for the polar study, is a change in ataxia as measured by ICARS, or the International Cooperative Ataxia Rating Scale. And in the next slide, I'll describe it in a little bit more detail. And then, of course, you're always going to be looking at safety. We're looking at other measures of ataxia as well as motor function. We're looking at strength, cognition, and we always like to hear what the patients are saying and what the clinicians are saying. So we have scales that we ask them to fill out, and we'll show you some of that data on how they think overall they're doing on the trial. In terms of safety, in this trial to date, we have not had any serious adverse events. Of course, we've had adverse events in general. About 2/3 of the patients experienced at least a mild or moderate adverse event. And what we see mostly, that we monitor very carefully, something called an infusion-associated reaction, which is happening in about a third of patients. They're mild to moderate, and what these are just reactions to GLM101, typically in the first 1, 2, or 3 infusions. After that, we don't see them anymore, and it's just as patients are getting tolerized or getting used to GLM101, their body reacts a little bit to it in the beginning, and then they're doing fairly well after that. This is usually just managed by either stopping the infusion, giving a dose of an antihistamine or a steroid, and then patients usually would be finishing up their infusion and then they've gone home. So this is something we monitor and is probably one of the most common events that we see within the trial. So as I mentioned, the endpoint outcome that we're most interested in at this point in what we're seeing has to do with ataxia. So how do we measure that within a clinical trial in order to see if patients are improving? we use something called the ICAR scale. And this is in the guidelines now for PMM2-CDG for clinicians to use in general to monitor ataxia and the impairment associated with it. So what we're doing is we're looking at this scale. It has 4 different components to it. It measures how patients sit and walk, how their limb coordination is, meaning how are their tremors, the coordination of how they can move their arms and legs. There's a speech component, understanding how the fluency and understandability of the speech is, repeating a sentence, the same sentence throughout the study. And there's also an eye exam that's also part of it as well. So what's important to know is that the scale, it's scored from 0 to 100. So the higher your scale, the more severe your ataxia is. So if your numbers are going up, if your scale is going up, you're worsening in terms of ataxia. And if your numbers are going down, that means your ataxia is improving. So there's about 19 assessments within this scale. It's usually administered either by a physician or a physical therapist, and it takes about 20 minutes to perform. And what we saw when we did this in our natural history study, because we added this in when we were starting to understand that this was an important measure for patients with PMM2-CDG to monitor. You start where you start. When we started our natural history study, this wasn't in the guidelines. It wasn't thought as a clinical assessment. But when we realized that it became part of the guidelines, this is what clinicians were using, we added it into our natural history study. And when we look at participants over age 12, in the natural history study, what we were seeing is about a 2-point worsening in the ICARs over 6 months. So as I was saying, the more you are increasing your score, the more severe you're becoming. So what this is saying, if you see all the lines going up, is that these are patients worsening in their ataxia. And I could say when we look at the scores, because there's the 4 domains, where we see most of this worsening is in that kinetic ataxia portion of the ICAR. So that's the tremors and the coordination of the upper and lower limbs. When we look at what we saw for the ICARs for patients treated with GLM101 in the 002 study, we were seeing improvements. So again, the lower your score, the less severe you're becoming. So by week 24, by the end of the study, we're seeing about a 12-point improvement in the ICARs. And this was seen in all 4 domains of the ICAR scale. And this is really the data that we were seeing over the last year that really were making us very excited to see a clinical endpoint that was improving and something that we could measure. Now, because this is an open label trial, we needed to now have another trial to understand, well, what does this look like against untreated patients on a placebo, treated in the same way under the same conditions? And that's really what's driving the new trial and why we're doing it to prove this concept that we're seeing of an improved ICAR score and improved ataxia. But even beyond the ICARs, even beyond that endpoint, we're gathering information, as I said, on how patients and clinicians are assessing their change over time. And these are global impression scales that we ask patients, since the beginning of the study, how do you interpret, how do you feel you've done since then? And you could say either very much improved all the way down to very much worsened. And in asking both clinicians and patients, what we've seen over the course of the study is that almost every patient has said they've improved in some way, and no one has said they've worsened over time. And even beyond these scales, which you're just checking a box, we hear anecdotes from patients through the clinicians that they're improving even beyond what we can measure in the trial. Like they can walk better on uneven surfaces, like sand. Like over the summer, we had a patient go to the beach and really be able to maneuver the beach better. They're able now to walk up and down stairs with less support than they required previously, and a general reduction in tremors. Our assessment for the for the verbal part of the ICARS, it's just a sentence. But what we're hearing from patients is that they're becoming more vocal. They're using more words, more difficult words. Their processing speed is faster, which allows them to engage more with family and friends. It's giving them more confidence in the realm of school and even at home. And in terms of the eyes, we were saying that patients are now having better fixation of being able to look straight forward versus the nystagmus that some patients experience. And it allows them to be able to have a conversation and be able to, as they're using their new words, to be able to look the person more better in the eye and focus on them, during those conversations. We've also heard that there's reduced headaches, improved concentration. So these are all anecdotes that patients have told us that we can't capture, and we're not capturing necessarily on a form in the trial. But this supports what we're seeing and what patients are saying in terms of their improvement in the trial, as well as what we're seeing on a more qualitative assessment like the ICARS. And then finally, just to give you a sense of one of the items on the ICARS is called an Archimedes spiral. So patients will have a sheet of paper and they have to trace the spiral around. And what we're seeing on the left is a tracing of the Archimedes spiral before treatment. And then to the right on the screen is you're seeing it 12 weeks after treatment. And this, just to put it in context, would be a 1-point improvement on the ICARS scale. So these are the assessments we've done in the 002 open label. These are the results that we were seeing. And again, this is what really has drove us to now look more closely in that more gold standard, if you will, study where it's randomized, it's placebo controlled, it's in multiple different settings in different countries and different sites to really prove the concept that GLM-101 is helping in that ataxia realm, as well as potentially some other areas that we're looking into. And with that, I will hand it back over to Dr. Edmondson to discuss a little bit more about the POLAR trial and the 003 trial, and then we can open it up to questions.
Great. Yeah. So the POLAR trial is this next phase of studying GLM-101 and determining how generalizable the results that have been seen in the 002 study that Rose just described, those preliminary results from how much that can be generalized across different clinic sites, different patients, and the way to be confident that what we're seeing is really related to the GLM-101 is by having a placebo arm where the physicians and the patients don't know what's happening. All of those assessments are being collected and everyone is blinded so that those prospective reporting can really be then analyzed as whether or not the patient is actually receiving the medicine or whether they're just enrolled in the trial but not receiving the GLM-101. So we can go into some of the details of this. So it's overall about a year-long study. The first half, or those first 24 weeks, roughly 6 months, is the randomized double-blind placebo-controlled portion where patients who are enrolled in the study will come in to get the IV infusion, and it'll either be GLM-101 or it'll be placebo. And the teams are taking great care so that physicians and patients cannot tell which one they're getting. And then after those first 6 months on the study, everyone gets switched over into an arm of the study where everyone will be receiving GLM-101. And so this, for folks who have been familiar with some of the different PMM2 clinical trials that have been going on, is sort of a guaranteed switch over at that 6 months mark to get it. And the 6-month timeline is a guaranteed portion of the protocol. So everyone will be switching over at that time. Although just so everyone's aware, the blinding procedures will continue so that you don't know what you were on in the first half until they've been able to collect all of the data of the study. But everyone will, for the second half, be receiving GLM 101 and having the same assessments. So the study, based on those improvements and the statistical analysis of how much we think patients should improve if they're on it versus if they're on placebo, requires about 50 participants to be in the study, half of which will get the GLM 101 in the first half of the study, and half that will be receiving the placebo infusion. And that across all of the various international sites will be to get to a total of 50 patients, the youngest of which are 4 years old, and that's really driven by the reliability of being able to complete the ICARS. Now, we know some very severely affected patients with PMM2-CDG may have trouble following the directions of the ICARS, or may be so severely affected. And so patients, to be eligible, also must be able to fully complete all of the ICARS assessments and not have a score above 80, so very, very severe ataxia, but must also have enough ataxia that there'll be room for improvement. And so a minimum score of 20 on that 100-point scale is required. So that's part of the initial screening procedures, is to make sure that any patient who wants to be in the study is able to fully do everything that's in the ICARS assessment and be severe enough that there's going to be room to see improvement with GLM 101. As Rose said, the primary endpoint, so the way we're going to be able to say whether or not GLM 101 is actually causing an improvement, is specifically by measuring those changes in the ataxia scale, that ICARS that she went through. But because we want to capture all of the various ways people are improving, there's some other measures of ataxia that will be part of those assessments that are built in, including strength assessments and cognitive testing, as well as a lot of safety assessments to make sure that this continues to be safe, and those patient and clinician reported outcome measures of how they think participants are doing during the study. So that's the main overview there. In order to get those 50 patients and to get them as quickly as possible, we've learned from the prior studies in PMM2-CDG that you really need a lot of sites to do this, and you need to do more than just a single country-based study to get enough patients. And so 15 planned sites throughout the US and Europe. It has already opened, so there are a few sites that have already started to give GLM 101 or placebo infusions to enrolled patients, but hoping to get this the full 50 patients enrolled sort of as soon as possible. And the other thing I should mention specifically is the enrollment of those 50 patients is a little bit restricted when it comes to ages. So need to be at least 4 years old, but the study, in order to be generalizable across both children's and older individuals and adults, will be limited to only 14 PMM2-CDG patients under the age of 12 years. So there's further limitation there. And then we know it can be challenging to get to these sites. People don't always live super close to these sites, so there's a lot of travel assistance provided as well for making these weekly trips to the study site in order to get the infusion. And we're super excited that in the US, we have sort of 3 currently active sites that are ready to start enrolling patients. My site at Children's Hospital of Philadelphia in Philadelphia, Pennsylvania, Christina Lamb's site at Seattle Children's, and Dr. Ava Morava's site in New York City at Mount Sinai. And by the end of this month, we anticipate a fourth site for those folks who are living in the Midwest will be at Dr. Sarah Foglu's site at University of Minnesota in Minneapolis. So hopefully those are pretty well distributed across the country to not make too challenging to visit for families who are interested in enrolling in the study. And we'd love to answer your questions, and anyone who feels like they're excited and want to be part of this study, we recommend that you reach out, particularly if your CDG physician is one of those on this list as an investigator. You can reach out directly to them to schedule the screening and to start participating in the study. And if you're not familiar, your primary physicians are not listed on here, you can either reach out to any of us at these primary CPG sites, or you can send an email to Glycomine, to the info@glycomine.com, and they will connect you with the site that seems most convenient for you.
Great. Thank you so much.
That's the slides. So happy
Great.
to take questions.
Yeah, so we are going to take questions. If you feel comfortable enough to raise your hand and come off mute, you can do that. Otherwise, you can put your question in the chat, and I will be delivering them to either Rose or Dr. Edmondson, whoever can answer them.
I have a question.
Yes, please.
Jan Valentin from Denmark. I just saw a few some time ago that Novo Holdings were reporting, who is actually putting money into this trial, that they're reporting some of the initial results, but you didn't show any results here. So those results based on your trial, or don't you have any results to show us from the Polar study today? Because I'm a bit confused on where Novo Holdings got those results for this type of treatment.
I'm not really sure which what you're referring to, but Novo Holdings is part of our investors. But if I had, I don't know what you're referring to, but if I had, I'm going back into the slides, is that they were showing a change. It was probably in the ongoing 002 trial. The Polar trial just began, so we really only have baseline results, and we won't have results unblinded to the public until the entire part A is completely done. So there won't be any results until it's all finished, for at least up to the first 24 weeks.
All right.
So if you saw results, it would have been from, my guess would be from from the 002 trial.
Oh, okay. And unblinding will happen in what, a year from now or something like that?
It would be after the last patient is finished, after week 24. So if I can go back to the design, sorry if I'm making people dizzy. So the part A, that's the randomized double-blind portion. Once we lock this data, so that's part of the process, is we finish, the last patient has their last assessments at week 24. We make sure all the data is in there, that it's accurate. We lock the data, means you can't change it after you lock it, and then we run the statistical analysis on that data, and we're unblinded at that point, and that's where we would be, having the top line data, I would say, from the part A, and that is the primary endpoint of the study, is after part A. So it would be after that. So it depends on where we get that last patient in, will determine when we get to have those analyses performed.
Okay, so but you will stop enrolling, and when you got the last patient, or will you also have, or will you stop by the end of 2025, even though you don't have patients?
Well, we are anticipating that we will be fully enrolled by the end of 2025.
Okay. Thank you.
Yeah.
All right, there is a few questions in the chat. The first one is, I get why it's reserved for ages 4 and above in order to get better observable measurements. Would there ever be consideration to try younger patients when traditional milestones are being reached?
So yes, once we are finished and we have a better understanding of the outcome of this trial, the next trial is planned for younger patients. So this trial would go up to age 4. The next trial would be down. Again younger. I'm as young as potentially 6 months to age 3. And as we understand some of the outcome measures, some of like the gross motor function, we're learning about what we could look at from an efficacy standpoint, and then to also obviously continue to understand the safety in that younger population. So that's the long answer is what I said. The short answer is yes, there will be a younger study on the heels of this one.
All right, another one in the chat. Hi, I'm a parent of a 2-year-old boy with PMM2 CDG from Slovenia. My questions are following. Is there an ETA on when GLM101 will be released to patients with PMM2 CDG that are not included in the trials?
That would be determined by regulatory approvals, depending on what jurisdiction you're in. Whether you're in the US, that would be an FDA approval. If you're in Europe, it would be an EMA approval. If you're in the UK, it would be an MHRA approval. So it would come after we complete this trial. We have to put that whole package together, as I mentioned earlier, and we submit it for review. So once we would get the approval, then that's when you can start the steps of being able to, have your physicians write that prescription.
Yeah,
And
just
then
to tag on to that, I would say these regulatory processes are long, particularly for a brand new therapy that's never been approved for any indication. And so it is a sort of a longish timeline to get that, but it's not something that's available outside of a trial, or that will be available outside of a trial unless it gets regulatory approval to be used.
we need lots of approvals. Okay, is there any ETA when phase 3 will begin?
So we will see what happens with this trial, and what this data shows us, and we'll plan accordingly. Yeah.
Has this been done with anyone unblinded?
I'm not sure. So the 2A trial, the 002 trial, all unblinded. This trial has a component of blinded and unblinded in the part A, and then everybody becomes unblinded in part B. There were other studies, as Dr. Edmondson was involved in, that were blinded studies as well. But I could let Andy, you could speak to those, to the acetazolamide, right?
I could say, so we did the acetazolamide trial that was, again, a blinded placebo control. That study ended, and then we unblinded everyone who had been blinded. And I think the reassuring things from that are that in the blinded phase, looking at a very similar measure of ICARS and ataxia, on the folks who were on placebo, did not have sort of a significant placebo effect improvement in the ataxia. So like Rose showed here, for the patients who are on open label, so not blinded, to get their GLM101 in the 002 trial, where they have those improvements in ataxia, in our acetazolamide study, we did not see folks reporting that improvement. It looks much more like the natural history data, where the ataxia stays or slightly worsens over time. And that study was done in a very similar population of PMM2 CDG patients as far as same ages. So we allowed over age 4 up to adults. And the majority of the patients in that study were adults with PMM2 CDG. But yeah, I think maybe the answer to the question as well is that these results that were shown were all in patients who were receiving GLM101, not blinded, with that improvement of ataxia. The Archimedes spiral improvement was someone who also received GLM101 and knew they were receiving GLM101. And those reports of the improvement over time, that was all in clinicians and patients who knew that they were receiving GLM 101. And the safety assessments and all of that has also been very closely monitored throughout the study in patients receiving this.
All right, great. So a few questions about the maintenance after the 6 months and 1 year, as far as what they will have to do. Do they have to get weekly infusions, daily? Like, can you just describe a little bit of what happens after that as far as the maintenance for the infusions?
Yeah, the second phase of the trial, the second 6 months, is the same as the first 6 months. It's still weekly infusions. The blinding will look the same. The sort of major assessments of the ICARs, those outcome measures will also be at the same frequency. So sort of every 3 months for those major assessments at the 6 month mark. So it just continues the exact same. And at this point, the anticipation is that any ongoing therapy after approval and things would be a weekly infusion dosing regimen.
How often are the ICAR assessments taken?
Every 3 months. Except right at the beginning, you're going to do an ICARS as a screener. You're going to do another ICARS just a couple weeks later before you get that very first infusion for an official baseline. And then 3 months, 3 months, 3 months, 3 months.
Great. All right, we have another question. We are excited for our two oldest daughters to start this month with Kiki in Minnesota. She has a 1.5-year-old and is excited to get her in when she's older. She would like to know what has helped patients get through the weekly infusions.
Karen, I can't see who's on the call. Do you know if Dr. Serrano or
I believe that she has not joined. Yeah, I think
Okay.
I'll speak on that. I think what really will help patients with the weekly infusions, one is the care team. They're going to get such great care going through this, but also just to get as a parent, your child, what will keep them entertained and comfortable for a period of time. So whether I know Dominic would definitely need a fully charged iPad, maybe a blanket, something to drink. So I think that it's just about making them comfortable and making sure that their care team is making them comfortable too, which is always good. I hope that answers your question. Okay, so let's continue. All right, in the 2A study, what were the most frequent non-local reaction adverse events? And what is the understanding of GLM 101 and the improvement in neurological function without crossing the blood-brain barrier?
So those are good questions.
Yeah.
Let's bring it back to the safety slide. So probably after the infusion-associated reaction is probably the most common one that we've seen in terms of adverse events. We've also seen probably the second most common is a mild to moderate increase in cholesterol in some patients, not in all patients. Some patients requiring some treatment for it, most patients not. And in terms of, actually, can you repeat the exact question for the second part for the blood-brain barrier? Yeah.
Yeah, so what is the understanding of the drug and the improvement in the neurological function without crossing the blood-brain barrier? Yeah.
And that's exactly right. The drug doesn't cross, GLM-1 does not cross the blood-brain barrier. And we have been thinking about this for some time of how what that mechanism is of why we're affecting some of these central nervous system events like the ataxia. And what we've come to understand is that GLM-101, although designed to deliver that mannose 1 phosphate within the cell, it probably works differently within the periphery, so outside of the brain versus inside of the brain. And what we are, understanding is that there's actually a component that mannose itself, which GLM101 also delivers a level of mannose to the body, not just mannose 1 phosphate, that is likely impacting the way the nervous system is responding to the ataxia. We're still working out that mechanism. We only have animal models to work with, obviously, to try to sort this out. But what that's been the understanding that we've come to over the last, say, six to 12 months is that there's a different way the drug works within the central nervous system versus the periphery. So multiple mechanisms of action that the body's experiencing.
Yeah,
That's a
I
very
mean, that
simple
really,
answer. I don't know, Andy, maybe you can. I
the
know
other
we
thing,
did it.
even simpler,
Yeah.
that maybe is in the periphery as well. This is probably going to lots of different organs, including muscles. So this could be improving muscle strength, which can also have some impact on, like an ICARS assessment, these motor assessments. So it may be that it's, improving muscle strength, as well in the periphery. So, anything really that is going to give a functional improvement to the lives of PMM2 CDG patients, almost regardless of how it's doing it, as long as it's safe. But if it's effective in making patients more, functional, have less care needs, be able to live their lives in a happier, more engaged function, then we're excited about that as a treatment possibility. And so if it's improving muscle strength, I know a lot of our patients have low muscle tone, low muscle strength, and so we'd love to be able to improve that as well.
Great. If you could just say what the age limit for the 003 trial is again. Somebody missed it.
Yeah, over age 4, no upper age limit. And as I said, only 14 of the 50 participants will be under age 12. So we are anticipating that the sort of pediatric under age 12 arm or portion of the study is going to probably fill up the first. But then anyone over age 12, sky's the limit. Oldest patient I'm aware of is, late 70s, eligible.
Okay, so we have another question in the chat. Hi, I live in Kansas and being part of the trial in Minnesota is not possible. Is there a way you can partner with our local children's hospital so my son can be a part of the trial?
Yeah, unfortunately, that's not the way clinical trials work, where you can receive like the therapy in one institution and then be assessed in another, or even, have all of it be done within an institution that isn't on the list at this point. It actually takes quite a bit, as I think Dr. Edmondson knows, to open up a site. And this is just where we went in terms of the expertise. I know you're from Kansas. I was not close. We, like Dr. Edmonds said, we do support travel, even if it's a plane ride, if you're willing to do so to any of the sites. But I think unfortunately, opening up a new site would actually take probably 6 months at this point if to add a site. So it's not possible.
All right, just a few more. Assuming GLM 101 makes it through all of the approvals in the US, could my child then receive the infusions with a local physician clinic instead of going to one of the sites?
So the answer to that is yes. That once it's approved and it's available, then it's up to your treating physician to prescribe it and sort how and where the patient would receive it. Can say from a long-term perspective, sort of thinking, once it's approved, the thought is that this wouldn't even need to happen in the constraints of a clinic, that home infusions could be a possibility after, per se, you say like the first few infusions you see, you do it within the constraints of a clinic under supervision. But once those first few infusions are kind of done, the long-term thought is that this could be an at-home infusion that you just hook up to once a week, and then you see your physician at regular intervals.
Great. And we have one more in the chat. Has this been tested with kids with sucrose and fructose intolerance? And if so, how did the body react, or what was the side effects?
So currently, of the 27 patients that we've treated, none of them had those issues. And I think Dr. Edmondson, I think you asked us about this as well for a potential patient, and we didn't think it would be a problem in terms of the treatment.
Yeah.
All right.
Has not been studied in anyone with those others. Really in totally healthy adults and in adults and children with PMM2-CDG, but not additional sort of specific health concerns.
Yeah.
Okay. Is there any evidence so far showing improvement in ocular health?
So on the ICARS, we are seeing improvement on that scale. We are doing more in-depth visual assessments, and that data is being analyzed now as we're finishing up the study. So what we do see is improvements on the ICARS.
On in the O2 study, are you doing this is a question for me.
Yeah.
Are you doing like retinal eye
Yep.
exams and things to be able to see if you see anything there? I know this is a concern for some of our, adolescent and adult patients developing the retinopathies.
Yeah.
And how that might be. And certainly that's also part of this study. There are eye exams at baseline and then at 6 months and at the end of the study to also be able to look at that in a blinded fashion. So that'll be exciting from the O2 to hear if there's anything obvious that's been seen there.
Yeah.
Yeah,
So
thank
I
you.
would say more to come.
All right. And then we have two more in the chat, and I think we'll stop it there. So I think this is an easy one. Is this going to be a lifelong drug once approved?
Yes, because it's basically substrate replacement. It would be a weekly infusion, and if you stop it, you'll likely stop seeing the positive effects.
All right. And then the last question, if my child participates and shows improvement, what happens after the study is over? Can they continue with the medication, or is the medication withdrawn until FDA approval? It's a good question.
Right. So right now we have an additional study called an open label extension. If you'd like the number, this is 003, that number is 007. And we will continue that study for participants who complete the 003 study and both themselves and their clinicians perceive benefit and they'd like to continue, they will be offered access. And it's still within the constraints of a trial. It's the way we continue to collect the data, but it's more of a safety trial than anything else. And so the assessments and the what needs to be done is much less than sort of the more intensive, say, the phase 2, 3 trials.
I think we will end it right there. We have 4 minutes to spare. Thank you so much for everyone that joined today. I hope this helped answer all of your questions, clarified some of your questions. A big thank you to Dr. Marino and Dr. Edmondson for taking the time to be here with us today. I'm just going to do a quick plug for our community. Our next CDG Community Connection will be on October 7th. So if there's anything you'd like to talk about, if you need some support, you can join us either at 11 a.m. Eastern or 7 p.m. Eastern. We'd love to see you there. And have a great weekend. Thank you so much.